here. And so, okay. And she also had some of these poorly defined areas of defects in her bone where there was a hypodense region in the middle and some hyperosteotic areas surrounding them. Uh-oh. Oh, there we go. Okay, so the pediatrician who saw this person was also a geneticist and decided to do whole genome sequencing. And this is what came out. So the patient was heterozygous for a T to G transversion nucleotide 1940 in the Lamin A gene. And that resulted in an amino acid change, usually a leucine, to arginine residue 647. And the mutation was de novo, as it wasn't present in the mother or father. And we know they were their mother and father because they shared approximately half of their rare polymorphisms with the patient. So this looked like a spontaneous mutation right here in the LMNA gene. And this is when the pediatrician and the genetics people called me because they said, gee, don't you know something about this gene? I said, well, yeah, I know a little bit about it. So this takes us back to 1993 when we first characterized this gene. And sort of the basic structure of the gene is shown here. It has 12 protein coding exons. Actually, there's another 1A exon within this large intron here that encodes for a germ cell isoform that we missed at the time. You see the gene encodes two proteins, Lamins A and Lamin C. Lamin A and C are identical for most of the way, and Lamin A has, or it's actually Prelamin A, 98 unique amino acids, and Lamin C, 6 unique amino acids. And soon after we did that, we collaborated with Jeannie Lawrence back in 1995 to localize the gene to chromosome 1q21.2. So we now know in the human genome that there are 3 genes encoding proteins called nuclear lamins. There's LMNA that I just discussed. It's on chromosome 1. It encodes Lamin A, Lamin C, this minor isoform called Lamin A delta 10, and the germ cell isoform called Lamin C2. There's another Lamin gene, LMNB1, on chromosome 5 that encodes Lamin B1 that's expressed in virtually all somatic cells. And there's Lamin B2 gene on chromosome 19 that encodes Lamin B2 that's expressed in all or most somatic cells, and a germ cell specific isoform. So let me just give you a brief history of what are lamins. And this probably most of you know, but I'll just go through it quickly. So at least my history of the lamins that I was taught back in the 70s, Günter Blobel and people in his lab purified a fraction of rat liver nuclei that they called the lamina pore complex fraction. And this is an EM of that purified subcellular fraction. And this fuzzy stuff on next to what's the internuclear membrane are lamins. And you can see the outer nuclear membrane that still has ribosomes on its outer surface, or maybe a few ribosomes, and a pore complex. Back around later 1970s, Larry Gerais, when he was in Günter's lab, purified or partially purified these 3 main proteins that were in rat liver nuclear envelope preps. He made antibodies against them, and they called these proteins Lamin A, Lamin B, and Lamin C. About 5 to 10 years later, Uli Aby and Bob Goldman purified these proteins, or to cut these proteins out of gels, or purified them as best they can. They only were soluble in things like 8 molar urea. They dialyzed away to urea, and these proteins formed these paracrystals that were reminiscent of intermediate filaments. And then in the middle 1980s, this is around the time I first started working on this stuff, Uli Aby with Larry Gerais took this now classical picture of a lamina fraction from Xenopus oocytes and showed that the lamina resembled cytoplasmic intermediate filaments. They estimated these filaments to be 10 nanometer diameter. And cDNA cloning in Günter's lab and in Mark Kirshner's lab showed that the nuclear lamins were homologous to cytoplasmic intermediate filaments, that they shared primary and secondary structure. So now forward a few years into the future, And this is work from Ohad, who maybe is on here today. He did cryo-electron tomography of the nuclear lamina and showed that actually the lamins are more like atypical intermediate filaments. They're probably not, at least in these somatic cells, 10 nanometer diameter, but closer to 3.5 nanometer diameter. And you see our beautiful reconstructed image of the nuclear lamina with nuclear pore complexes. This is the inner nuclear membrane, the outer nuclear membrane, and this is pretty much our picture of the nuclear lamina today. So intermediate filament proteins that are different in some regards from their cytoplasmic counterparts associated here with the internuclear membrane. Ooh, that was strange. Okay, so now we know that's a little bit of the cell biology, and we move in, and it started with work that Giselle did. I remember getting a call from her old mentor, or a friend of her old mentor one day saying, oh, we found a mutation in the Lamin A gene that correlates with a disease that is associated with a disease called Emery-Dreifuss muscular dystrophy, which is a cardiomyopathy with skeletal muscle involvement. And the first thing I said was what Günter Blobel told me when I was in the lab. He said, that's impossible. Mutations in lamins would be incompatible with life. Although it turned out that wasn't impossible, and certain subset of mutations cause muscular dystrophy and dilated cardiomyopathy. Soon after, a generally different set of mutations in the same gene were shown to cause a lipodystrophy, where the patients lose fat from their extremities and they have other problems of insulin resistance and complications of that. There's a peripheral neuropathy was shown to be caused by a specific homozygous amino acid substitution in the Lamin A that causes Charcot-Marie tooth type 2 disease here. And then, maybe I guess these were last discovered, was a rare group of diseases that really were multi-system. They led to growth retardation and features of progeria like micrognathia, skin modeling, other things I can't see on my slide. And we're going to focus on this group of diseases today. So the classic of these diseases is Hutchinson-Gilford progeria syndrome, and everybody here probably knows it. And at least according to the internet, this is Hastings-Gilford's patient, one of the persons who the disease is named after. And you can see this is actually a young kid. I think this kid is in his teens, probably in his teens. And, I dress him up here with his cane. He looks like an old man. And if you look at him here, you could see some of the major defects of the disease, the growth retardation, the bone deformities, these pretty significant joint deformities, this small jaw. And what you can't see in this picture, and what ends up really killing these people at young ages, is the changes in the vascular smooth muscle that leads to obstruction of coronary arteries and cerebral arteries. And these kids usually die from stroke or myocardial infarction and complications at a fairly young age. So Prelamin A is a precursor to Lamin A. So we talked about Lamin A and Lamin C, and those are the proteins along with the B-type lamins that make those filaments. But Lamin A is actually made as a precursor pre-Lamin A. And this protein undergoes a series of processing reactions that leads to Lamin A. So as most of you know, the enzyme farnesyl transferase catalyzes the addition of a farnesyl group to this cysteine that's triggered by the sequence CAAX here, in the case of Lamin AC, CSIM. Then an endoprotease chops off the last 3 amino acids, the AAX, if you will, or the SIM in this case. Then an enzyme called isoprenyl cysteine carboxyl methyltransferase catalyzes the carboxymethylation of the cysteine. And then in the case of Prelamin A, this metalloproteinase, the MPST24, recognizes this farnesylated tail and cleaves it right here between this Y and L sequence. And this piece is chopped off and it disappears, And again, it's dependent on the first step. It has to be, protein has to be farnesylated for this to happen and this to happen, and then for this to happen. And this Prelamin A is really gone, and what incorporates into the lamina is Lamin A. And after all these years, this was figured out sometime in the 1980s, I believe. Nobody really knows the reason for this processing. But we do know what happens if this processing doesn't happen. And the first example here is the Hutchinson-Gilford Progeria syndrome. And this disease is caused primarily by a single mutation, which is a nucleotide change that creates a cryptic splice donor site in the middle of exon 11 of the LMNA gene. This causes an aberrant splicing event where the resulting protein is a truncated protein that goes from what's encoded by the middle of exon 11 here. It has the sequence of exon 12, which includes the CAX box, which is farnesylated. But this truncated protein is lacking the ZMPSD24 cleavage site. So these patients retain, they're heterozygous patients, they retain a truncated, permanently farnesylated Prelamin A that was called Progerin. And I see the people are on my side here. If I move them, this is an immunoblot from a patient that we saw several years ago. And you can see these are cells from a normal patient. You see Lamin A and Lamin C. Lamin B1 was on the blot. And in the patient with Progeria, you see this protein migrating in between Lamin A and Lamin C. That's this protein, Progerin. So there are other diseases. I hate to show this picture, but I think, we should all see to some extent what we're working on. This is a disease called restrictive dermopathy. That's really a disease that's not compatible with life. These babies die right around the time of birth. And in these cases, these are cases where there's a total loss of function of the ZMPST24 protein. And this is work done by Steve Young and collaborators looking at cells from one of these unfortunate babies. And you see that these people, will just look here at these columns, these babies only express Prelamin A. There's no Lamin A. The Prelamin A cannot be processed because there's total loss of function of this enzyme. They have Prelamin A, Lamin C, no Lamin A. This is an antibody specific for Prelamin A. But some cases are milder that have mutations in ZMPSD24. And these have been variably described as mandibuloacral dysplasia or an atypical progeria. And in these patients, there's some residual ZMPSD24, usually compound heterozygous mutations with partial enzymatic activity retained. And you can see here, this baby has some Lamin A, mostly Prelamin A and Lamin C. This is work from Sue Shackleton. This described this case. And you can see in this person, the very prominent bone defects. You can see here, the tips of the fingers almost aren't there. You can see on the x-ray again, the acral osteolysis and more space between the bones than normal. And you can also see again the nail dystrophy and the mottled skin, similar to what the patient we found has. And this is work from Susan Michaelis, who showed a few years ago that if you take different ZMPSD24 mutations that are found in different patients, there's a pretty much good correlation between the amount of enzyme activity and the severity of the disease. Turn that off. And as you can see here, wild type, this is they actually did an assay in yeast to measure the ZMPSD24 activity. If you call wild type 100%, you see these patients with decreasing enzyme activity. It tended to correlate with the degree of disease, ranging from mandibuloacral dysplasia to a more severe Progeroid type phenotype to total loss of function, restrictive dermopathy, and neonatal lethality. So this is pretty much describing defects that you can have in Prelamin A processing. So, or some of them, you look at wild type, there's the CAX box, it's farnesylated, the AAX is cleaved off, it's carboxymethylated, and you get mature Lamin A from processing. In Progeria, you get a deletion of the ZMPSD24 site, so you get retention of this farnesylated carboxymethylated Prelamin A variant called progerin. And in restrictive dermopathy or in mandibuloacral dysplasia, you have total loss of function of ZMPSD24 and you retain unprocessed Prelamin A, or you have partial loss of function and you have some unprocessed Prelamin A and some Lamin A. So let's get back to the patient that came into our institution a few years ago. And if we look at what's happening to her, she has an amino acid substitution right at the ZMPSD24 cleavage site. And research long ago from several groups showed that changing the amino acids right at this cleavage site prevent cleavage. And this patient had this arginine substitution here at the site. And you can see that it blocked ZMPSD24 processing. And in the bottom of the slide, oops, I think I did something. In the bottom of the slide, you can see the immunoblot from the patient. The patient had about half Prelamin A and half Lamin A. She was heterozygous for the mutation. And here's an immunoblot for Prelamin A showing she had a lot of Prelamin A in her cells. Whoops, where am I here? The Zoom is giving me a hard time. Okay, so I think I lost a slide here. Anyway, what we recently did, and this is work done mostly by Yusho Wang, who's on the call here, we generated a mouse with the corresponding mutation. And these mice, the homozygous mice, were L648R, L648R mice, and you can see one of these mice here. If you look at these mice, I think these are homogenates a liver from these mice. These are wild type mice. There's no Prelamin A. In the heterozygous mice, there's Prelamin A, and you can see about roughly equal amounts of Prelamin A and Lamin A, maybe a little more Lamin A. And if you look at the homozygous mice over here, these mice express only Prelamin A and no Lamin A, just like the patients with the restrictive dermopathy. So just to go on with the characterization of these mice, we watch their growth over time, and we see that the heterozygous mice essentially grew in the same rate as wild type mice. But if you looked at the homozygous mice, they weight gain leveled off at a few weeks of age, several weeks of age, and their weight actually began to drop as they got older. But one thing, if you look at here, most of these mice were still weighing them out here at almost 2 years of age. So even though they're homozygous for this mutation and they're making only Prelamin A, they're living a relatively long time. And this just shows the mice again, and you can see as they get older, and again, quite old, the mutant mice are smaller than their wild type counterparts. And we did some analysis of body composition, and we showed that these mice had, I guess you can call it to some extent, a generalized lipodystrophy. They had decreased body fat. This is at maybe 7 or 8 months of age. They had decreased body fat per total body volume. And when we went, though, and looked at insulin resistance, they were not insulin resistant. So they had this lipodystrophy, but not with some of the consequences of a severe lipodystrophy. So we followed these mice out for as long as they lived. And now I think Yusha has followed them out till all the homozygous mice were dead, which was well over 2 years. And you see this actually a sexual dimorphism. The male mice die sooner overall than the female mice. But even if you look at the male mice, 50% are still alive at 90 weeks. Whereas with female mice, there's over 50% are alive out here to over 100 weeks. So these mice, despite again making only Prelamin A and no Lamin A, live quite some time. Okay, so what first thing we did is we wanted to say, what, let's compare them to other similar mice that are out there in terms of longevity. And this is probably one of the best mouse models for Hutchinson-Gilford Progeria syndrome, the LMNA G609G mouse. And these homozygous mice, you could see here, this was work done by Steve Young and Lauren Fung. These mice are almost all dead by about 20 weeks of age. If you look at the ZMPSD24 null mice, and at least except for a single amino acid substitution, they look the same as the L648R mice. They make only Prelamin A and no Lamin A. These mice are all dead in 24 to 28 weeks. And again, if you look at these L648R, L648R mice, these mice live, many of them, to over 2 years. So, a question becomes, what is the difference between these and these and these? Okay, so we went and phenotypically characterized the mice in a little more detail. And what I'm basically going to show is a series of slides that they basically almost exactly phenocopy the ZMPSD24 minus mice, and to a large extent the HGPS mice, except that the phenotypes are slightly milder and they come on at later ages. So this is a reconstruction of the zygomatic arch of normal and mutant mice. And you can see here that the zygomatic arch at 4 weeks of age is mostly normal. But if you look at these same mice at 30 weeks of age, in the wild type mice, the zygomatic arch is mostly normal. But if you look at the mutant mice, I just have to move everyone. Okay. If you look at the mutant mice here, you basically see this degeneration of this area of the zygomatic arch, which I believe is near where one of the sutures is. And this is a little bit reminiscent of the acral osteolysis. This is really resorption of the bone as the animal ages a little bit. You see it's born with a pretty much normal zygomatic suture here, but as it ages, in these wild type mice, it doesn't change. In these mutant mice, you get resorption of bone. If we look at the mandibles, the jaws of the mice, these here in wild type mice, they have these very well developed mandible. And you see in the mutant mice, this is a CT reconstruction, you see there's really degeneration of the mandibular bone here. And these are male mice and these are female mice, wild type, mutant. And this is really the same thing, looking at it a different way. You can see these different processes of the mandible. This is how they look when they're normal. And in the mutant mice, you can see degeneration of these mandibular processes. This is just a more quantitative analysis of the changes in the mandible. These mice are around 7 or 8 months of age. You can see here a normal mandible from a male mouse and a female mouse, the front and the back. And you can see again here in the mutant mice, the resorption of the bone from the mandibular processes. And if we quantitatively measured these distances between these different parts of the mandible, you see they're all significantly smaller in the male and female mutant mice compared to controls. These mice also had this dental malocclusion, which is seen in the HGPS and the ZMPSD24 minus mice. At young ages, it's not that severe. As the mice get older, it becomes quite severe in many of these mice, and almost all the mice had this dental malocclusion. It's a result of these changes in the mandible. Again, thinking of the girl we saw, small jaw and problems with her teeth. And just anecdotally, what we found, and this is interesting, some of these mice, if we treated their mouth, if we shaved their teeth in some way, I don't know, some veterinary technician did it, they shaved these teeth, I believe, and the mice actually started to gain weight when they were put back in the cage. And a lot of the weight loss they have, maybe as they get older, maybe secondary to failure to eat food because of this dental malformation. The mice also have a generalized osteoporosis phenotype. We looked at bone density of the spine and we measured it quantitatively. If we look at male mice and female mice, this is just images of the trabecular bone of the vertebrae. You see significant differences in the bone density of the male and female L648R homozygous mutant mice. And this is just some CT images of the legs, lower legs here. And I think you can see pretty just qualitative differences between here and between, say, here and here. Cardiovascular phenotypes, as you know, in HGPS, this is the really lethal problem that kills the children. And mandibuloacral dysplasia, it's less clear. There's been so few cases. There's only been a case report or two of people dying from early cardiovascular disease. But we're in the process of performing a more advanced, sophisticated cardiovascular evaluation. I could say when the mice are around 8 months of age, their cardiovascular system appears to be normal, but we have some preliminary data that when they get much older, they may begin to get some of the changes in the vasculature that HGPS mutant mice get. Okay, so most of you know that one sort of readout for laminopathies, and particularly the progeroid laminopathies, is alteration in nuclear morphology. So we isolated embryonic fibroblasts from these mice, and you see the mutant mice express only Prelamin A in their fibroblasts, wild type mice only Lamin A and no Prelamin A. When we looked at these labeled for Lamin AC or labeled for Prelamin A and looked under the microscope, you can see these abnormal shaped nuclei here. I have some better pictures, but we're just showing these here. And the number of abnormal shaped cells with abnormal shaped nuclei increase with passage number. You can see at 3 passages, maybe about 30% had abnormal shaped nuclei, and increased to maybe 60 to 70% with more passages. Now, again, many people have shown long ago that in cells from patients with their mice with restrictive dermopathy, the ZMPSD24 minus mice, or the HGPS mice, or patients with HGPS, these nuclear shaped abnormalities are partially reversed by treatment with a farnesyl transferase inhibitor to block the farnesylation of either Prelamin A or Prelamin A variants. And you can see here when we treated cells from these mice with the farnesyl transferase inhibitor, these are probably after 5 passages, the number with abnormal nuclei went from around 70 or a little over 70% down to about 50%. So similar to what's been seen in these other progeroid disorders with Prelamin A processing defects, there's an FTI reversible abnormal nuclear morphology. And we showed the same thing in dermal fibroblasts from the patient with the corresponding mutation. Again, an increase in abnormal nuclear morphology with passage number, and treatment with FTI partially restored the abnormal, restored normal nuclear morphology. This patient is heterozygous. She had, this is the opposite way, is percentage with normal nuclear morphology. And you see with FTI treatment, the number with normal nuclear morphology was similar in patient as control, as composed with no treatment. There were more with fewer with normal nuclear morphology. Okay, so again, just touching on treatment, I think everybody may know this, but you know, pioneering work really was in ZMPSD24 minus mice, and then later mice with a HGPS mutation showed that FTIs improve phenotype and prolong survival. This was work done by Steve Young and Lauren Fong. I got their names wrong here, but that's okay. Based on this and based on a few clinical trials that were done on patients with HGPS and a few patients with ZMPSD24 mutations, the US FDA just last year approved lonafarnib for Hutchinson-Gilford progeria syndrome and what they call on the label processing deficient progeroid laminopathies. And, we wonder, although we haven't done this yet, if FTI will have the same, sorry about that, I think we're having an earthquake here or something. FTI would have similar effects in the L648R mutant mice. And should we try it in the patient, although our patient has become lost to follow-up, she hasn't come back to Columbia. Does everybody hear that noise? They're tearing down a building across the street. All right, I'll keep going. So I'm near the end. This is just a set of conclusions here. So deficient pre-Lamin A processing, as we all know, leads to progeroid disorders. The LMNA L647R and the mouse L648R mutation blocks pre-Lamin A processing. A human subject with this mutation accumulates pre-Lamin A and has a relatively mild, at least compared to HGPS, progeroid disorder. And the homozygous mutant mice, that should be an 8, not a 7 there, that express only pre-Lamin A, for some reason have a much milder progeroid phenotype and live much longer than the corresponding ZMPSD24 null mice. So this raises several questions that I think we and others should look at. Why is the patient that we found, a young woman, less sick than a heterozygous patient with HGPS? One's making a truncated variant, one's basically making pre-Lamin A with a single amino acid substitution. Why are these homozygous mice less sick than the ZMPSD24 minus mice? Again, they're both expressing only pre-Lamin A with no Lamin A, but these have lived much longer and have less severe phenotypes. And I think a couple questions or a couple possibilities come up, questions to answer. Some research has shown that ZMPSD24 has other functions, for example, in translocon unclogging, also in mediating certain antiviral defenses. Might these mice have defects that, in addition to some toxic amount of pre-Lamin A, causes them to get sicker sooner? Is it possible that the single amino acid substitution alters some activity of pre-Lamin A? Could pre-Lamin A with one single amino acid change in its tail be doing something different than the normal pre-Lamin A? And another question, is there less pre-Lamin A in cells expressing this variant or this variant pre-Lamin A than the normal pre-Lamin A in these ZMPSD24 minus cells? We don't think so based on when we look at it compared to expression of other things, but you never know if it's some problem with different avidity of the antibody or something else. And this brings up one more last question what we have, and I took this picture from a paper we did with Raphael and with Ohad. We looked at the nuclear envelope of cells from a different mouse with a different disease. This is a mouse that Giselle made long ago, the LMNA H222P mutant mouse that develops cardiomyopathy and muscular dystrophy, not progeria. And using the cryo-electron EM, we saw some changes in the lamina, not so much in the filaments themselves. The filaments appear to be mostly normal, but changes in the way that the chromatin was packaged around the filaments and the expression of different lamin genes and proteins and heterochromatin proteins. So, now that we have cells from these mice, maybe we can take similar pictures and see how cell expressing only pre-Lamin A and no Lamin A, how the lamina is affected in the structure of the lamina, the nuclear envelope may be affected in those cells. So I said I'd keep this short. I'm going to stop there. I just want to say 80% of the work that I talked about today was done by Yusha, actually the characterization of the patient and the mice. I think she's on with us today. And other people in the lab and other collaborators at Columbia who's helped out in our work with progeria and progeria-related diseases. And this work was also done in collaboration with Susan McAllis at Johns Hopkins. She's an expert on Prelamin A processing. Wei Su at Harvard is an expert on bone and bone development, who helped us with some of the characterization of these mice. And Peter Nagy is a friend who used to be at Columbia, now at Praxis Genomics, who interpreted the genomic analysis of the patient and will help us with some other work going forward. So that's it. And like I said, I wanted to keep it short and have the rest of the time for discussion on this or anything else.